Hi Jacob, at long last I'm making the video for how to color, how to fill out and color the DNA origami template for you. Okay, so that goes with, of course, the DNA activity. I'll need this color code, and I noticed on your Jamboard that you made this sequence of DNA, okay? And what I want to start with is this upper right hand corner base. First base in your sequence is adenine. All right, and then going to the left, the next one in the sequence would be guanine, then adenine, and guanine, and so on. So you're alternating adenines and guanines. All right, well, I want to fill out the template according to that sequence. So here I have my template. This is the top, the space that has some space on it's the bottom. At the top, in the left, is where that first base would go. So if I turn it this way, it's going to kind of match Jamboard that way. So that first base was the one in the upper right. So I want to put an A in there because your first base was an adenine, and adenine starts with A. And your next base was a guanine, and that starts with G. And adenine, and guanine, and so on, right? All the way down your sequence. If you don't have enough bases on your Jamboard for the whole page, that's okay. Just put in whatever you want at the bottom. When you run out of bases, just put in either A, G, T, or C, up to you. Now, I've got the top filled out. I've got to do the bottom also. And you noticed when you were making the Jamboard DNA model that adenine pairs with thymine. So thymine's going to go across from adenine, but it's going to go across from adenine diagonally. Okay? Let me turn that so you can see it. Adenine pairs with thymine, okay? So then guanine, G, pairs with cytosine. Adenine pairs with thymine. Guanine pairs with cytosine, and so on. The reason that I'm doing this on the diagonal is because when you finish coloring and folding this, it's going to be a two-sided, three-dimensional structure. I'll show you an example of that in just a second. So that's why we're putting the complementary base diagonally across from what it matches up with and not horizontally across, okay? Leave this top one blank because that's going to be how we hook yours onto the bigger strand. Okay, so once you've filled out this side, okay, all of it, and then you fill out this side, remember I said this is going to be a two-sided thing. So the right-hand side of this template is going to be a mirror image of the left-hand side. So when I fold it, it's going to be the same on both sides. So this middle column, right next to where I put that T, it's going to be the same. It's going to be a T. And right next to where I put that C, it's going to be the same. It's going to be a C. And T and C, just like that, okay? And then this far right column, okay, is going to be the same as over here on the far left. Those are going to be mirror image of each other. A, G, A, G, and so on all the way down, okay? Once you've got that all lettered, okay, then you're going to color it. Now, how do you color it? Well, let's look back at our color code again. It says on our color code that adenine is orange, thymine is green, cytosine is yellow, guanine is purple, okay? So adenine is orange. I'm going to need an orange colored pencil, and I'm going to color wherever there's an A for adenine. I'm going to color that orange. It's very important that you color the right part, though, so I'm showing you how to do that now. You color this parallelogram shape where the A is, the adenine. Color that all in orange, okay? All right? That's what you color in. Okay, not this rectangle, the parallelogram shape. Okay, the parallelogram shape. All right, and so wherever there's A's, you're going to color those in that way. Wherever there's A's, that's what you're going to color in. Those parallelogram shapes, A's. Okay, I forget the color code, so I got to go back to that again. Sorry. Thymine is green, cytosine is yellow, guanine is purple. Okay, cytosine is yellow, guanine is purple. So wherever there's a G, I'm going to color that parallelogram purple. Okay, purple. That goes there. Color in that parallelogram. Sorry for my sloppy coloring. Okay. Cytosine is yellow, so that's a C. Wherever there's a C, I'm going to color that in, that parallelogram in, right? Yellow. And thymine is green, so I'm going to color that parallelogram in green like that. So that's how you color it. And when you're all done coloring it, okay, both sides are going to be colored. Color it correctly, all right? And then you're going to fold it so that when you're done folding it, it's going to make a three-dimensional DNA molecule, okay? So you can see I've got my adenines, which are orange, matched up with my thymines, which are green, and my guanines, G's, are purple, matched up with my cytosines, which are yellow. So yours, when you're done coloring it, you're going to fold it, so we're going to add it to our chain. So far I've got everybody else's on. I'm going to add yours down here at the bottom. All right? So that's how you color and fill out and color the template. There's a separate video for how you fold it. I hope these instructions are helpful, and good luck.